So, welcome to the UK-ICN seminar series. We're down to one speaker today, I'm afraid, because of a diary clash for the second person. But we still have the pleasure of having Rachael Dempsey from Julian Hiscox's lab in Liverpool, who's going to talk to us about G2P in MERS. So, with that, Rachael, over to you. You're good to go.
Yeah, good, thank you. So as Paul said, I'm Rachael. I am a third year PhD student in the Hiscox lab, and I'm going to talk to you about my PhD project, which is genotype to phenotype consequences in MERS. Oh, 1 second. Oh, my slides won't move. 1 sec. Okay, there we go. We'll just have to do that. So MERS is the most severe human coronavirus known, and the World Health Organization reports an approximately 35% mortality rate. It was first identified in Saudi Arabia in 2012, and the majority of the cases since then have been in countries in the Middle East, with a few cases or outbreaks popping up in other countries as well. The symptoms are very similar to those that you would get with SARS-CoV-2. They include fever, cough, shortness of breath, pneumonia, and GI symptoms. And again, similar to SARS-CoV-2, the severe illness can result in respiratory failure and can leave patients requiring mechanical ventilation. And it's also elderly patients and those with pre-existing chronic conditions that are at the greatest risk for developing the severe disease. So MERS is a zoonotic virus. It actually has a high genome relatedness with bat coronaviruses, but camels are the major reservoir. And transmission-wise, camel to camel, camel to human, and human to human transmission have all been confirmed. The cases that have been seen in humans do tend to be those that come into close contact with camels. And similarly, when we've seen human to human transmission, it tends to be within households or in healthcare settings. And then also more recently, MERS has also been identified in domestic animals, such as cattle, donkey, goat, and sheep, that are in close contact with camels as well. So just briefly on the biology, because I'm sure this audience will be familiar. MERS is a positive sense single-stranded RNA virus, and it has a 30 kb genome, which is just shown across the bottom of this slide here. And the genome contains a large polyprotein that's processed into 16 non-structural proteins, the structural proteins and the accessory proteins. And as we know, the spike protein allows entry into cells, and it does this by binding to the DPP4 receptor. And as is the case with other coronaviruses, the spike protein is variable, and any mutations could potentially affect immune escape, transmissibility, or pathogenesis. So why MERS? So as I've already kind of said, MERS causes sporadic infection in humans. It's the most pathogenic coronavirus, but currently there's also no therapeutics to treat MERS. There are some vaccines and therapeutics kind of in development, but nothing like readily available at the moment. Recombination and genomic mutations are common across all coronaviruses, and any sort of genotype changes like this could alter the biology of MERS disease, which obviously means there's pandemic potential there. And it's also just important to improve our understanding. So can we identify any sites that could contribute to increased transmission or immune evasion? And then finally, there has been some work done on MERS, but a lot of this has been mainly focused on the EMC 2012 strain, which now is obviously 12 years old. So there's been some changes since then, and hopefully we can have a look at some of those mutations. So the hypothesis for my PhD project was genotype to phenotype consequences in MERS may cause different pathogenesis. And I have two main aims. So the first is to generate MERS spike pseudotypes to characterize genomic mutations identified in clinical samples from Saudi Arabia. And the second is to generate a full-length MERS clone to study virus biology and evolution. So for the first part of my talk, I'm going to concentrate on this first aim. So as we know, coronaviruses have variability in their genome, and MERS is no different. So this panel across the top here is just showing, it represents an alignment of more than 500 clinical samples. And the kind of red lines that you can see are showing the variability across the MERS genome. So where the red lines are larger, these are sites that are more variable. So as expected, we're seeing quite a lot of that variability in spike. But what is less well known for MERS is what these like mutations and this variability, how this could be impacting things such as viral load, patient outcomes, and transmissibility. So for this part of my project, as I mentioned, I was using pseudoviruses. So obviously I have concentrated on mutations of interest that are occurring in the spike. And I've listed these mutations that I selected in this table here. And yeah, some of them we really don't know very much about. Others, such as this amino acid 411, this residue is known to impact neutralization. These that are highlighted in yellow here are residues that are known to directly interact with the DPP4 receptor. And then in grey on the right-hand side, these residues are close to the S1, S2 cleavage site. And so these were some of the reasons why these were mutations of interest that I've selected to have a closer look at. So the pseudoviruses that I've made, they are based on a lentiviral-based system. And this pseudotyping system was very kindly provided to us by Dr. Dallin Bailey's lab. And it consists of these two plasmids, so one with a 5' luciferase reporter and the HIV-1 regulatory genes, and then one with HIV-1 GAG-POL core genes. And then these are co-transfected with the MERS spike expression plasmid into HEC293 T cells. And then this generates MERS spike pseudotypes, which I've then used to look at entry and neutralization. So the so the wild type that I've listed in this table here was kind of like the starting point for my pseudoviruses. And this sequence was based on a clinical sample that is from Saudi Arabia and was isolated in 2019. This was very kindly given to us by our collaborator, Mohammed Al-Sayer in James Stewart's lab. And it has 5 spike amino acid changes versus the EMC 2012 reference strain. And then to generate the other mutants that I've used for this, I just did site-directed mutagenesis of this spike plasmid. And I've included a schematic of spike over here just so you can kind of see where the locations of these mutations would be. They're mostly in the receptor binding site or close to this S1, S2 site here. So the first thing I looked at was the wild type pseudovirus confirmed to be functional. So I just made the one wild type spike sequence pseudovirus initially. And what you can see here is the Western blot, which confirms expression of MERS spike, the HIV-1 GAG-POL lentiviral core, and also the 5' luciferase reporter as well. Then I did a luciferase-based infectivity assay, which is in this panel here. So the MERS pseudovirus is in the kind of lilac purple sort of line. VSVG, which is known to pseudotype well, was used as a positive control. That's in the red line. And then the negative control is with no glycoprotein. So just an empty vector was transfected alongside the pseudotyping system for this. So the kind of titers in this luciferase assay were about 1,000-fold higher for the pseudovirus than the negative control. Then I had a look at receptor binding of this pseudovirus. This was using a split GFP fusion assay, which is shown in this panel here. And this is where spike and one half of GFP is transfected into one cell type, and the DPP4 receptor and the other half of GFP into another. The cells are co-cultured, and if the GFP comes together, hopefully you'll get some GFP signal, which we're seeing here, showing that the spike and DPP4 are binding, and we're not seeing any for VSV or the negative control, which is as expected. And then finally at the bottom is microneutralization, and this is showing that the pooled patient sera is able to neutralize the MERS pseudovirus. So once these were confirmed to be functional, I then continued to generate the mutants. And what I'm showing here is the infectivity assay for those mutants. So this is the same luciferase-based infectivity assay. And what I found was that these mutations that I've introduced don't seem to impact infectivity in this assay. And the mutant titers seem to be comparable to the wild type. So the wild type is still in that sort of purple color, which is kind of here. So the results for the infectivity assay were fairly comparable. I then did the split GFP fusion assay, and this showed that I was seeing some differences in receptor binding in this assay. So I've just pointed out mutants 7, 11, and 8 here, because these mutants were showing better receptor binding than the wild type, which is again in the purple line just here. And then mutant 7 had the mutation I529T. Mutant 8 had E536K, and mutant 11 L745F. And then kind of at the other end of the spectrum was mutant 1, kind of had the most reduced receptor binding in this assay, and that had the mutation T387P. So the next thing that I wanted to look at was neutralization of these different mutants. So to do this, the pseudoviruses were incubated with pooled patient sera for an hour before being added to HEC293 T cells that are expressing DPP4. So as you can see, mutant 2 seems to be less well neutralized than the wild type. So mutant 2 is in the orange here. And this mutant has the L411F substitution. And if you remember, this was the mutation that has kind of previously been shown to impact neutralization. So it was nice to see that I was getting the same results here as well. And then mutant 7 and mutant 9 seem to be neutralized the most efficiently. And they had I529T and W553R substitutions in the spike. So the conclusions for this section are that the infectivity of MERS pseudovirus mutants was comparable. I529T, E536K, and L745F substitutions resulted in improved receptor binding when compared to the wild type. However, the mutant that was carrying I529T was neutralized more efficiently by pooled patient sera, and also the L411F was neutralized least efficiently by pooled patient sera. So that was kind of a summary of where I'm up to with the first aim, and now I'm going to talk a little bit more about generating a full-length MERS clone to study virus biology and evolution. So this just shows kind of a summary of the method for making the MERS full-length clone. And this work was done in collaboration with Professor Andrew Davidson's lab at the University of Bristol. And they had previously used this method to generate SARS-CoV-2 full-length clones as well. So just briefly, to start with, I generated overlapping fragments that spanned the whole MERS genome. These were then assembled in yeast. The yeast clones were then screened, also sequenced, and then once they were correct, they underwent in vitro transcription. The RNA was transfected, and then hopefully virus was recovered. So this is kind of showing the overlapping fragments that I generated for the full length clone. So there was 12 fragments in total, and I generated fragments 2 to 11 by RT-PCR. And this was using RNA from the same clinical sample that was used to generate the pseudoviruses in the first part of my talk. The end fragments were actually designed and synthesized using GeneArt. This was so that they had overlaps to the yeast vector that they were being assembled into. And then the assembly was done by transformation-associated recombination in yeast. So once they were assembled, I needed to screen the yeast colonies to see if they had assembled correctly. So this is done by designing primers that target the areas that are where the fragments overlap. And when I did this, I found that the 3 yeast colonies that are shown down here were all correctly assembled. However, when they were sequenced, only 1 out of the 3 had the identical sequence to the starting material. So this was the one that was chosen to undergo in vitro transcription. So the full length and end gene transcripts were transfected into Vero cells, and then the MERS full length clone recovery was confirmed by IFA, and that's what's shown in this image here. So in green is double-stranded RNA, in purple is the M protein, and blue is the cell nucleus. And double-stranded RNA is a hallmark of coronavirus replication, so this confirmed that it's successful virus recovery. And then Ed, who's one of Andrew's postdocs, very kindly did some growth curves of the MERS clone to see how it compared to the EMC 2012 reference strain. So these are just rescue replicates of the same clone. They should all be the same. And the replication, for the most part, was fairly comparable. Apart from this clone 2.1 at 48 hours appears to be impaired. I then went and sequenced these and found that there were some changes in the spike N-terminal domain, and that could be, could account for the differences in the replication kinetics that we're seeing here. So the changes that I found in the N-terminal domain of spike in the full-length clone was G94R, Q98R, and Q304R. So what I did was I introduced these into a pseudotyping system to see if I could see any changes in infectivity, receptor binding, or neutralization. So this is the infectivity data over here, and you're seeing some differences, but they're fairly comparable to the wild type. The wild type does look like it's performing kind of slightly better. And then in this panel is the split GFP fusion assay, and this is where we're seeing some much bigger differences in the receptor binding. So all 3 of these mutations seem to have a negative impact on receptor binding when compared to the wild type. However, this is what I've been doing for the last couple of weeks. This is literally just 1 replicate, so I do need to repeat this and make sure. And then again, this is the microneutralization data that I have so far. It is just 1 replicate as well. And it looks like the mutation, that the pseudoviruses that are carrying these N-terminal domain mutations are being neutralized more efficiently by the pooled patient sera. But again, this is just 1 replicate, so this needs to be confirmed. So to conclude this section, MERS full-length clone was successfully generated by adapting a reverse genetics method that had previously been used for SARS-CoV-2. The full-length clone was assembled correctly in yeast and the sequence confirmed, and then the virus recovery was confirmed by IFA. Replication of the MERS clone was comparable to the EMC 2012 reference strain, but there were some differences observed between the rescue replicates. And then sequence changes were identified in spike N-terminal domain, and that may account for those differences that were seen in the replication. And then my preliminary data suggests that the N-terminal domain substitutions negatively impact receptor binding but I need to do those experiments a few more times before confirming this. So for next steps, I'm going to continue characterizing the pseudoviruses, which have these N-terminal domain mutations, continue characterizing the reverse genetics clone, just waiting on a risk assessment to be signed off for that. And then introduce mutations of interest identified using the pseudotyping system, which I was showing you at the beginning of this talk, into the reverse genetics clone for characterization. And yeah, with that, I'd just like to thank my supervisors and everyone in the Hiscox lab and the collaborators. And if there are any questions, I would be happy to answer them. Thank you.
Oh, thank you very much, Rachel. Congratulations on getting the reverse genetics clone to work too.
That was very, very much a team effort with Andrew and Ed as well as postdoc in Bristol.
I'm going to abuse chair prerogative here. Did it just work first time?
Yeah, the virus was recovered first time. So when making the yeast clone, the sequence wasn't correct straight away. Had to go back and kind of do repairs to that. But yeah, the first sort of attempt at virus recovery, this was done down in Bristol by Ed and Andrew, and yeah, worked the first time.
Okay, that's what I wanted to ask. It specifically was, did you find how much were you plagued by mutations in the yeast cloning step? Because that's what we're seeing here.
Yeah, I think if I remember correctly, the first attempt, there were kind of like changes everywhere, and then went back and repaired those. And on that second attempt, after doing a repair, we were able to carry on, and it was correct.
Okay. Questions from others for Rachel? You can either type it in the chat or just turn your microphone on.
Paul, I've got one.
Yeah.
Rachel, hello. So I should know this. So the 5 mutations that you identified in Mohammed's sequence,
Yes.
which are different from the Erasmus strain.
Yep.
Do you have all 5 of those together in one spike, or are they all separate?
The 5 that are different in the clinical sample versus the Erasmus strain, they will be in all of the pseudoviruses because
Right.
the base of pseudoviruses on the clinical sample.
Right, okay.
Because we weren't sure if we could make an Erasmus one when I first started the work.
Yeah.
We weren't sure if I could use that sequence as well.
Yeah. Thank you.
That's okay.
Any more questions? They're all being shy. So the GFP assay that you're interpreting as being receptor binding.
Mm-hmm.
Talk me through how you normalize that. How do you know it's receptor binding per se and not like efficiency of lentivirus production and so on?
So it's not done. I may not have explained this clearly. I'll go back because I had a diagram on one of my slides. It doesn't use the pseudoviruses. It's just by transfecting the plasmids. Where's the slide? This one.
Oh, okay, so I misunderstood it. Sorry.
So no, sorry, I might not have. So it is into the effector cells. It is the different spike plasmids, whichever one has whatever mutation, plus a plasmid that has half of the GFP. And then the target cells is the other half of GFP in a plasmid and the receptor expression plasmid as well. And then they're incubated like not together, separately, for 48 hours. And then after 48 hours, they are co-cultured. And then if the spike is able to bind the DPP4, the GFP should then be complete and glow green. So I haven't included, there was probably just already too much on this graph, but I haven't included the VSV and pcDNA on this graph. But when you do it with those, you don't see any binding. You only see binding if the spike is able to bind to the DPP4 receptor.
So the assay actually measures everything from expression to cell surface transport to fusion, but
Yeah.
you know the mutations are in the receptor binding domain, so that's the likely explanation. Okay.
Yeah, yeah.
Nice. Well, I will carry on asking questions as nobody else is popping up. So the neutralization resistance assay, the 411,
Yes.
do you see any sign of a selective advantage of that in the population of MERS sequences? I mean, is it becoming more predominant, more prevalent?
I think it's quite hard to say. So there isn't an awful lot of sequences available. So when I was doing alignments, it kind of seemed like an awful lot of, well, there were not an awful lot, but there were quite a few sequences available from, say, like 2018, 2019. And then during the COVID pandemic, there just really isn't any MERS sequences. So the only few that I could get hold of were very kindly provided by Mohammed Al-Sayah in James's group, whose PhD was included sequencing some MERS viruses. But yeah, I think that mutation seemed to crop up. I think it was around 2015 when there was the outbreak in Korea. And it did seem like it was definitely increasing in the population, but there's really not that very many sequences to be able to tell for sure.
But is it independent selection within a particular infected person, or is it? Are you seeing it in transmission, and does it work in camels as well?
I'm not sure about transmission. I think so. It didn't seem like it was like kind of coming up as a minor variant. It was kind of from about, I think, 2016 onwards. We've seen it a lot more in like the sequences. I'm not sure about camels though.
Okay. Okay, there's a question appeared in the chat from Aya Donovan Banfield. No microphone. Are any of the NTD mutants in equivalent residues to what's known for neutralization evasion in SARS-CoV-2?
I don't think so. I was trying to have a look at this last week actually. So I couldn't really find anything on these residues, and they didn't seem to marry up with the ones for SARS-CoV-2. But yeah, I'll have to see, have to do the assay again because I've literally only done it the once so far.
Okay, well, if there's no more questions, let's just congratulate Rachel again on a great talk, beautiful data, and thank you very much.
Thanks very much. Thank you.
Okay, short one today with just one good talk. Thank you to the audience for turning up as well, and see you next time.